Urban stormwater runoff is one of the largest contributors of contaminants to Puget Sound. Stormwater runoff is definitely an indicator of what's happening in our air pollution as well. The really exciting thing about the research that we're doing and the results that we're getting is that it gives people hope. Green stormwater infrastructure really can be part of the solution. My name is Dr. Jenifer McIntyre. At Washington State University, I study urban stormwater runoff and its impacts on aquatic animals, and I am an innovator. Thank you. I'm a little abashed. That was so awesome. Hope I can live up to that. So, good afternoon, and today I will be sharing with you our research on the toxicity of urban stormwater runoff and the role of green stormwater infrastructure in controlling that toxicity. So very importantly, although this is a WSU event, this research is highly collaborative and conducted primarily between WSU, U.S. Fish and Wildlife Service, and NOAA Fisheries. And our funding primarily is coming from EPA Region 10, but also with important grants from other national, state, and nonprofit agencies. The reason that urban stormwater runoff is a problem is because this is not a forest, right? And because it's not a forest, it doesn't act like a forest when it rains. Water has nowhere to go. It quickly moves off of these hard surfaces, usually into the nearest water body, like the video just said. So here you're seeing a time-lapse video of runoff entering Puget Sound. This is what it looks like. Now from the surface, it looks like this, and here you're seeing the increased turbidity that's coming from particulates that are washed off of those hard surfaces. What you can't see is the complex cocktail of chemical contaminants that enter the water as well and can impact the health of aquatic animals. I'm going to talk a lot about coho salmon, so I want you to understand a few things about them if you don't already. We study them as a sentinel for impacts of stormwater runoff because they live where we do, in the lowlands of Puget Sound in this region. They're also very sensitive to both water quality and quantity. And unlike many of the Pacific salmon, they spend at least 1 year, more than 1 year in freshwater in small streams. Coho salmon return from the ocean at 3 years old. Their offspring then spend 1.5 years in freshwater before they migrate back out to the salt. So fully they spend half of their lives in freshwater, and they're vulnerable there to the impacts from stormwater runoff. I'm going to give you 2 examples of the integrated impact of stormwater runoff on coho salmon from field studies that I've been involved with. The first is an example with adult coho salmon. In 2002, the city of Seattle began monitoring adult salmon use of restored urban creeks. So the map is showing a variety of urban creeks that have had restoration efforts, and then Fortson Creek at the top is a pristine system that we used as a control. So one of the streams that the city started monitoring was Longfellow Creek, and they began to notice something there, that there was something wrong with the adult coho that were returning there to spawn. So we, including a large team of NOAA scientists, started studying the fish in these creeks. We noticed a series of behaviors in the spawners following rain events that rapidly progressed from lethargy and disorientation to eventually a complete loss of equilibrium, immobility, and even tetany, complete rigidity prior to death. Now this leads to high rates of premature mortality. Across years and across sites, we see that more than 60% of the adult salmon returning to these systems die before they get a chance to spawn. Premature mortality like this can be caused by other factors like elevated temperature that can cause problems with the amount of dissolved oxygen in the water, or it can exacerbate diseases like in the Fraser River sockeye salmon. But this is different. This is related to urban stormwater runoff. I'll come back to that in a few minutes here. The second example I want to give you is during embryo development. So at Longfellow Creek in West Seattle, we experimentally raised coho embryos in creek water that was either unfiltered or had been filtered through a classic system of sand and activated charcoal. So the top photo, you can see how that filtration affected the appearance of the water. Now in the actual experiment, we're taking creek water directly out of the creek, filtering it or not filtering it, and then passing it over the embryos as they develop. After about 50 days, we saw that the embryos in the filtered runoff had developed fairly normally, with only about 10% of them dead or abnormal in some way. In contrast, the picture at the bottom is of an embryo that had been developing in the unfiltered, just the regular creek water. So they were developmentally delayed. You see that that fish is smaller, and that happens when an animal is spending a lot of energy trying to detoxify its body. There's also a high rate of mortality and various abnormalities that focus mostly around the vasculature. So cranial hemorrhaging is what that arrow is pointing at, blood spots in the heads of the embryos. This is one more view of that, top down, looking at that fish's head, those fish's heads. In the top picture, I want you to see the beautiful symmetrical pattern of the vasculature in the head region of the fish. And in the bottom image, that you don't see that pattern. Instead, you just see some random blood spots. So definitely abnormal. All right, I will also come back to that in a little bit. So 5 years ago, I started working at Washington State University and took the lead on these stormwater studies. And one of the first things we did was move it from strictly in the field to also looking at in the laboratory in more controlled settings. And one of the things we wanted to do, we wanted to know what is in urban runoff, do very, well, at the time seemed very thorough chemical characterization. How toxic could that runoff be to aquatic animals? And is there anything we could do to prevent that toxicity? We chose to work with highway runoff initially because it represents a concentrated example of urban stormwater runoff, and roads are common to all urban land uses. This particular road, some of you may recognize, it has an AADT, that's the average annual daily traffic of more than 60,000 cars. And we chose this location not just because it was a busy road, but also because, as this arrow denotes, there's a downspout from this road directly into the parking lot at NOAA Fisheries. So that made it very convenient to collect runoff when it rained. In addition to coho salmon, I've been using the Zebrafish research model. This is a small freshwater fish that's been used extensively in human health research. The embryos are optically clear. You can actually watch blood moving through their bodies. They also develop rapidly. The heart starts beating at 24 hours. They hatch at 48 hours. Their swim bladder inflates sometime before 4 days. Very importantly, they respond toxicologically very similar to humans. So in terms of how chemicals affect their development and their organ systems, particularly the heart and the cardiovascular system. And then finally, their genome has been fully sequenced, so there's a lot of molecular tools that we can use. Oops, that's the development stuff. All right, zebrafish are much more hardy than salmon. They're not nearly as sensitive to toxic insult. But with zebrafish, we can easily study sublethal effects of runoff. And those include, across different storm events, general inability to hatch or a delay in hatching, developmental delay, so again, smaller size because they're spending energy to fight those contaminants or detoxify and eliminate them. There's a small eye phenotype that develops that I'll talk about a little bit. And then pericardial edema, so the yellow arrow is pointing to swelling around the heart. There's this fluid accumulation. This also happens in humans, for example, with congestive heart failure. And then finally, in more extreme cases, we'll see these deformities of the craniofacial deformities, and also we could see deformities of the heart itself. Oh, my battery's running low. If anyone wants to help me plug in the surface. Meanwhile, I will continue if I can. Wait, I have to say close, I believe. All right, I'll just continue and you let me know if I need to stop, okay? All right, I want to show you what some of these cardiovascular abnormalities look like. So the fish in the top row are 2 days old, and the zebrafish in the bottom row are 4 days old. Now, fish have a 2-chambered heart. It starts out as a tube, forms 2 chambers, and then for the rest of development to proceed normally, those chambers need to loop in a very particular way. These videos, the first ones here we go, are control fish, so exposed just to clean water. Now they have a normal heart, and you can see, hopefully, the 2 chambers beating and pushing blood through the bodies. The other pictures are of fish that have been exposed to different stormwater runoff events, and they're just examples of the types of abnormalities that we see. So here we have the pericardial edema, that fluid accumulation around the heart. Pooling of the blood, pooling of the blood before it enters the heart. It's not able to enter the heart as it should. The cranial hemorrhaging, like with the coho embryos, bleeding in the head region. And finally, in those extreme cases, actual deformities of the heart itself. Now, the kinds of cardiotoxicity I'm showing you, we know that they can be caused by cardiotoxic PAHs, so polycyclic aromatic hydrocarbons. And we know that there are PAHs in stormwater runoff. One way to look for exposure to PAHs is using the gene CYP1A. This is an enzyme that metabolizes PAHs so that the body can eliminate them. When an animal is exposed to PAHs, this gene gets turned on, and more enzyme is produced in the cells that were exposed. With antibody staining, which is what this is, we can attach a fluorescent protein to that CYP enzyme. So the green staining in this photo is individual cells in the skin of the fish that have this detox enzyme present. Now with this type of microscopy, using a confocal microscope, we can also focus in a very narrow region. It's like we're taking a slice through the animal. So if we take a slice through the middle part of the animal, it looks like this. You still see that green staining from the enzyme in the skin of the fish. And then we have attached a fluorescent red protein to the heart tissue, to the muscle tissue, and here it helps us see where the heart is. So if we zoom in on that, what I want you to see is, again, that line, that curve where you see the enzyme in the skin of the fish, but also now individual cells inside the heart that are showing this detox enzyme. So what we're able to see is that these chemicals are not just getting on the outside of the fish, they're getting inside the fish, and they're actually getting inside the heart. And we have also found heart-specific genes that indicate when there's injury to the heart. And we see that those also get turned on in fish exposed to runoff. All right, now I want to take you back to the field. In 2012, we wanted to know whether we could recreate the symptoms of pre-spawn mortality in otherwise healthy coho by exposing them to stormwater runoff. So to do this, we worked with the Suquamish tribe, who were very generous in being open to letting us kill their fish. The work that we did took place at Grover's Creek Salmon Hatchery, and that's out near Indianola, Washington, on the Olympic Peninsula, or Kitsap Peninsula, actually. And it receives coho, chum, and chinook that are returning from Miller Bay. Here's a picture of that. So to do this research, we collected road runoff from our location in Seattle, brought it out to the hatchery, and then adult coho were placed in these large PVC tubes, and they had water and air flowing over their gills, and then they were exposed either to clean water or to the stormwater runoff. So what we found is that after just a few hours of exposure, control fish, here we've taken them out of the tubes and we're looking at their behavior in clean water so that we can see them well. And the control fish are acting normally, swimming around the tank, a little agitated perhaps, having been confined. In contrast, the stormwater exposed fish are definitely showing signs of the symptoms that we see in the field after a stormwater event. So loss of equilibrium, and this animal is not very far from death. This fall, for the first time, we took a close look at what was going on in the blood of these fish. So these are blood smears that have been stained to identify different types of cells. Most of what you're seeing here are red blood cells. They have a pale cytoplasm and then a dark nucleus in the middle. But in the first slide, I have circled what is an immature red blood cell. It's smaller and has a darker cytoplasm. In coho exposed to runoff, we saw many more of these immature red blood cells. And those are released by the body when there's a low oxygen condition to try to get more oxygen moving through the fish's body. In the middle photo, there are 3 white blood cells. Now, in a healthy fish, it's very rare to run across a white blood cell. They're about 1% of the total blood cells. So to have 3 in one picture like this wouldn't happen. But in the stormwater exposed fish, we see much higher rates of these white blood cells present and that suggests that there is an immune response taking place in the fish exposed to runoff. Finally, the third picture shows a field of red blood cells with 2 thrombocytes. These are the flattened cells with not a lot of cytoplasm. Now, thrombocytes are the fish's version of platelets, so they're involved in blood coagulation. Now, what we see in fish exposed to runoff is fewer thrombocytes, and this suggests that the thrombocytes are actually falling out of the blood because there's perhaps microcoagulation happening in the blood in the fish's circulation, so small coagulation perhaps lining all of their arteries. A new tool we invested in this year to look at blood was a point-of-care analyzer. So this is an iStat. It's used in veterinary, but also in human health medicine, human medicine, to determine whether and sometimes how an animal is sick. That's all I was going to say about that. Very, very cool to have this level of results for the blood. So here are some of those results, and for all of these graphs, the controls are the white bars, and the fish exposed to runoff are the black bars. So this arterial blood gas panel showed, across 6 storm events this fall, a drop in pH, a drop in bicarbonate concentrations, a base deficit, and no increase in the carbon dioxide content of the blood. So if you got this result, your doctor would say that you're suffering from a metabolic acidosis. This can in fact kill you if it's left untreated. So it's a very serious condition. The blood chemistry results showed an increase in hematocrit. So PCV is a packed cell volume. It's the amount of a blood sample that is cells versus plasma. So for example, in the controls, about 37% of their blood was cells. But in the fish exposed to runoff, about 60% of their blood was cells. So this indicates that there's either an increase in the number of cells in the blood and/or a decrease in plasma. And this is a classic response to a lack of oxygen in the tissues. We also saw an increase in lactate in the fish exposed to runoff. And lactate is produced in cells under anaerobic respiration. So this happens when the body has to create energy in an environment without much oxygen. So again, suggesting hypoxia, not enough oxygen. Finally, we saw a drop in sodium. This is hyponatremia, which is a very serious condition that can be caused by diuresis, which is increase in urination, which can be triggered by hypoxia. And it can also be caused by a failure of the heart and/or the kidney. And those can in turn lead to hypoxia. So there's a lot of evidence that these fish, when they're exposed to urban runoff, are experiencing hypoxia, not enough oxygen in their tissues. So behaviorally, we see them with an increased gilling, increased ventilation, trying to get more oxygen into their gills, into their body. We see them swimming up to the surface and gaping, which fish do if there's not enough oxygen in the environment. Hematologically, we see that increased hematocrit, we see the metabolic acidosis, and we see the lactate production. So cellular hypoxia can occur for a variety of reasons. The simplest reason is there's not enough oxygen in the environment, which for fish is the water. But we measured that, and then we supplemented it when we needed to, so that wasn't the problem. In anemic or hypemic hypoxia, there's not enough red blood cells or not enough hemoglobin. And this can happen, for example, with carbon monoxide poisoning or something else that's binding to the hemoglobin, like in methemoglobinemia. Stagnant hypoxia happens when there's not enough blood flow to push that oxygen-rich blood to the proper tissues. That can happen when there's cardiovascular failure or hypotension, not enough pressure to push the blood around. Finally, there is histotoxic hypoxia, which is where the tissues themselves can't use or access oxygen. And this is, in fact, what we think that we are seeing, because we expect under histotoxic hypoxia to see an increase in the partial pressure of oxygen in this arterial blood. And that's what we saw with our fish exposed to runoff. So currently we have this hypothesized cause of death, which is not just scientifically pretty cool, but it's also useful in that we can now generate hypotheses that we can specifically test to help us identify, hopefully, what are the class of contaminants that can cause this and that are causing this from the runoff exposure. All right, now we don't need to know everything about how toxic runoff is or how it causes its toxicity to be able to try to do something about the problem. About 5 years ago, we started including in our research not just the problem side, but also what are potential solutions. One of those solutions is green stormwater infrastructure. So that's also called low-impact development. Now, green stormwater infrastructure, they're a series of technologies that allow the water to infiltrate into the soil, basically. And a lot of them include bioretention, which I have listed up there. And basically, that's getting the water to soak into the soil and using it not just to slow the runoff down, but also to filter out those contaminants. At the WSU Research and Extension Center in Puyallup, if you haven't been, there's a very cool, full-scale GSI research and mitigation project that includes replicated plots of permeable pavement, replicated plots of rain gardens where they're testing different plant palettes, and then replicated what we're calling mesocosms that have the same plants but are testing different soil types. Now, the research I've done so far is halfway between the lab and the field. It's using these large bioretention columns, like the ones that are shown here in our greenhouse. And I think of this like taking a core section through a rain garden. Most of it is bioretention media, and then there's some drainage layer at the bottom. And bioretention media tends to be very simply, in our region, recommended by Department of Ecology, a mixture of sand and compost. Pretty simple, right? So the first experiment we did looking at whether this was going to be effective at reducing or preventing toxicity from runoff, we collected the runoff in Seattle, brought it down to the center, and we mixed it all together, because at that time we were using glass carboys. Now we have a really fancy stainless steel tote that's yay big. And then we pulled off runoff that would then be our unfiltered runoff that we would test. The rest of the runoff went through the columns. Some of them had plants, and some of them didn't. And we collected the effluent that came through the columns then. And then we tested it on a variety of organisms, which included not just the zebrafish, but also that target organism, the coho salmon. So juvenile coho were exposed for 96 hours, and you can see here what some of that runoff looked like. Pretty dark, and you can also hopefully tell that the filtered runoff is not crystal clear itself. It picks up tannins from the compost, probably. Now, in the runoff, within 12 hours, actually, forget 96 hours, within 12 hours, all of those fish had died. In contrast, both in the clean water and in the filtered runoff, all of the fish survived the test, even at 96 hours. There wasn't a delayed mortality effect. Now, this is the same runoff that killed all of the fish in less than 12 hours, so pretty impressive. We were also looked at sublethal effects with that zebrafish research animal. So here we looked at whether the air bladders were inflated, what the fish's size was, whether there was that pericardial edema, so that would be a bigger pericardial area, and the fish's eye area. So the fish exposed to the untreated runoff, they had no inflated air bladders, they were smaller, they had fluid accumulation around their heart, and they had smaller eyes. But when that same runoff had been filtered through the bioretention system, with or without the plants, there was no added benefit at this point. It reversed that. So now they have normal air bladder inflation, normal size fish, normal hearts, and normal eyes, just about. There actually is a tiny effect still present there. We're not sure what that's about, but obviously a huge improvement. On the molecular level, we can look for that induction of the detox enzyme. So I'm going to paste over top here just what the fish is like so you can orient yourself here. We're looking at the top half side view of the zebrafish, and then on the bottom we're looking at a section of the tail. So in the control fish, all we see is that red stain associated with muscle tissue. There's no green stain from the detox enzyme. In the middle picture is the fish exposed to stormwater runoff. You see the detox enzyme in the skin. You see it inside the heart. And then in the trunk picture at the bottom there, you can also see it throughout the peripheral vasculature. And that's all of the veins and arteries running through the trunk muscles. In very sharp contrast, that same runoff that is passed through that bioretention filter, you can now see there's a little bit left over, a little bit of induction of this detox enzyme in the peripheral vasculature. You can see in the bottom picture, I hope there, just a little bit of green staining. But very importantly, none in the skin anymore, and the most important thing is there's not any induction anymore inside the fish's heart. And that's what goes along with the loss of that cardiotoxicity, no more fluid accumulation around the heart. So all of this research done, even just for this initial test, showed for different animals, different endpoints, almost all of the bioretention system had eliminated the toxicity, not just reduced it, but completely eliminated it. All right, now I'm going to bring our research back to salmon. This study we just finished in January and took place out at Grover's Creek where we grew coho embryos. And out to that system, we would episodically bring runoff from Seattle. This is, jump right to the end of the experiment, and this is survival at the end of that experiment. So we had fish that were in well water all of the time, all 65 days of this experiment. And then these other ones, we would episodically bring the runoff out, switch it from well water to either 50% runoff, 100% runoff, or 100% runoff that was first filtered through that bioretention system. And what you can see here is that fish exposed, even across we ended up doing 15 storm events, across 15 storm events to 50% runoff, had about 25% survival, even lower survival in the 100% runoff. But that same runoff, filtered through bioretention, we didn't see any mortality, effectively, no mortality. What about the adult coho? Will this bioretention system work for them as well? We saw that we could create pre-spawn mortality by exposing coho to urban runoff. Could we also prevent it by treating the runoff? So for this project, we built a bioretention system in 55-gallon drums that we could transport out to the hatchery. Now, following the Department of Ecology guidelines, we filled the drums with a 2-foot mixture of 60% sand and 40% compost, and then covered it with a thin layer of bark mulch. We helped collect the fish, and then we treated half of our runoff through the bioretention. Now, we expanded our previous experimental system to include that third treatment. We know that fish remain healthy in the well water. We know that they become symptomatic after just a few hours in the untreated runoff. What would happen in the treated runoff? Dun, dun, dun. So the first time we ran the test, we exposed the fish for just 4 hours. The controls were healthy. Half of those runoff fish were dead. Half of them were sick. And then the filtered runoff fish also appeared to be healthy. But we were concerned that the same symptoms would develop. It would just take a little bit more time. So all of the subsequent exposures were 24 hours long. And you can see now that the results are very stark, with fish in the treated runoff appearing completely unaffected after 24 hours, with all the fish in the runoff being dead and the treated fish looking just like controls. So again, a very impressive performance. Here are some videos to show what that looks like. It's dark now because it takes a little bit of time to filter the runoff. That top fish has been exposed to well water. Now we have the fish exposed to the unfiltered stormwater, which again is showing a very serious loss of equilibrium, which will end very soon. And then finally, fish in the filtered stormwater runoff, being again very active and responsive. So I want to go back now quickly to the question of how can we prevent toxicity from urban runoff? I've been talking about green stormwater infrastructure, which both reduces the amount of runoff and deals with the toxics that are present, but we're going to need more tools to solve this problem. Source control is another area that we need to focus on. So strategies already being implemented include street sweeping and education campaigns like the Puget Sound Partnerships, Puget Sound Starts Here, and the Department of Ecology's Don't Drip and Drive, which are great programs. And they're aimed at changing the actions of individuals. But then there's green chemistry, and this focuses on a process or a product and re-engineers it to move from chemicals that are known to be toxic in that process or product to chemicals that turn out to be non-toxic. So a research project that we've started is to look for the sources of toxics in the runoff. So in the road environment, we have toxics coming from tire wear, from brake wear, from the particles in exhaust, and from windshield washer fluid. And then we have the variety of automobile leaks that we end up having, from fuel, engine oil, brake fluid, and on and on. So which of these is going to be the most toxic of the items that get into the runoff? And then which of them contribute most to the toxicity we see in the road runoff? And this is going to hopefully lead us down the path to identifying where the biggest problems are, and then we can work on solving those. So, urban stormwater runoff, obviously a threat to aquatic ecosystems. We have seen in our studies multiple symptoms of toxicity in both fish, multiple fish species, also in invertebrates, which I didn't have a chance to show today. And we've also seen that soil bioretention can prevent, either reduce or completely eliminate that toxicity. Now, we have a lot of work to do. We've barely scratched the surface of what we need to know. Some of the outstanding research questions include, how long will these systems last? How long will they continue to perform this well? What is the effective sizing? How much do we need in a watershed to prevent something like pre-spawn mortality in adult coho? What's the optimal media to use? I'm not going to go through this list because it's quite long, but these are the upcoming research projects that we have going on right now at WSU Puyallup. And they involve a variety of technicians and graduate students, both at WSU, at UW, and at Evergreen State College. And we're very excited about what we'll learn in this next year or two with this research. So where do we go from here? How do we get where we need to be? What's going to make the biggest difference? These are some really important questions, right? I think John Stark is going to talk for a few minutes and hopefully address some of what I want to say about the Washington Stormwater Center. So that's the first thing I would do is point that out as a resource to people, both for education and for the research that we do, both for individuals, homeowners, businesses, and municipalities. And I also want to just quickly point out some things that are going on at the federal level. So these are two bills that are introduced into Congress in the last few months. The first one was introduced by Denny Heck, and the next one by Derek Kilmer. So there's the Puget Save Our Sound Act, which will hopefully provide more federal support for Puget Sound, which is a really important, very large water body. It should get its own focus. And then the Green Stormwater Infrastructure Act, Investment Act, which will provide incentives to help people implement the strategies that we know will work. So the idea is, with green stormwater infrastructure, with source control, even though we don't have the forest that we used to have in this region, we can help it to act more like a forest. These are, I'm not going to read them because, again, a very long list of all the wonderful help that we've had to do this research from a variety of people and institutions and nonprofits. And I hope that I have time to answer questions. Thank you.